Good afternoon and welcome to the webinar. I'm Candace Chapman, Director of Marketing for Genomenon, and I will be your host today. Dr. Mark Keel today will discuss characterizing targeted cancer therapies via a comprehensive gene fusion database. Possessing a comprehensive view of the research related to fusion genes can significantly accelerate the drug discovery and development process. This information allows researchers to find fusion pairs for any disease of interest, identify targeted treatment strategies, and refine fusion gene breakpoint analysis. Unfortunately, the diversity of gene pairs involved in fusion events can be overwhelming, and until now, there's been no comprehensive database of research citing gene fusions. In response to this need, we here at Genomenon have developed the most comprehensive database of gene fusion pairs found within the scientific literature. This is a very new development, and we're very excited to share it with you today. As you are watching, please feel free to submit questions to Dr. Keel through the chat window on the right. Mark will answer questions after the presentation. So without further ado, I'll introduce our speaker. Dr. Keel is a founder and Chief Science Officer here at Genomenon, where he oversees the company's scientific direction and product development. Prior to starting Genomenon, Mark completed his residency in clinical pathology at the University of Michigan. While at Michigan, he completed a fellowship in molecular diagnostics and devised the informatics framework for clinical next-generation sequencing in the molecular diagnostics laboratory. During his doctoral studies, he made groundbreaking contributions to the study of hematopoietic stem cells, for which he was awarded the Weintraub International Graduate Student Award and the ProQuest Distinguished Dissertation Award. While a postdoctoral researcher, he made significant contributions to the field of hematopathology, including genomic profiling of lymphoid malignancies, for which he was awarded the Benjamin Castleman Award. And now I'm going to turn it over to Mark. Mark, take it away.
Thank you, Candace. Let me know if I'm not coming in clear. And thank you especially to all the attendees. As Candace said, I will echo how excited we are to be discussing with you for the first time, at least publicly, a new development that Genomenon has undertaken using the Mastermind database, which is to say to expand our focus away from mutations and genetic data and disease associations to include structural variants, namely gene fusion events. So this is an outline for my talk. As an attendee of webinars, I like it when the speaker gets right to the point. And so I'll spend as little time as I think is necessary going over a background just to lay the groundwork for why this is important. Then I'll talk directly and immediately about what it is we're discussing so that you have a sense for that in your mind. And I'll actually show you some example data. I'll walk through a couple of use cases in outline form. And then I'll summarize the data and turn it back over to Candace to field any questions that we have. If we don't have enough time for questions, we'll be sure to answer them after the webinar. Before I start, I'll emphasize to anybody who's new to Genomenon that Mastermind is a software that's available to you through the link below. You can sign up here for 14 days of the professional edition, which is for clinical users. Large reference laboratories or academic clinical labs alike, but thereafter the database is available to you in its free edition. So this is one of, I think, 4 background slides that I have. This is from a recent Nature Reviews publication, and it's a little bit busy, but it's effectively a timeline of the discovery of and the meaningfulness of structural variants in the form of gene fusions in cancer. So in 1960, in the pink there, the first structural variant was discovered associated with CML, and then it took a decade or more for it to be more precisely characterized. And thereafter, you can see augmented by advanced technologies, including next generation sequencing, this great proliferation in our understanding of and characterization of fusion events that are oncogenic drivers in cancer. And so the way this is laid out is colored by those different techniques. And the review here was published not a year and a half ago, and I think they left off the last 5 years because it was just too crowded and there was just too much information across all of these different strata of different disease types, both in hematopoietic malignancies, novel fusion events were discovered, as well as increasingly in the case of solid tumor cancers. So apologize for the wordy slide. This is a review from a couple years ago now from a former colleague of mine, but I thought it captured very effectively many of the points that I want to convey in this introductory section of the webinar. So first is to say what I think is the headline is that gene fusions are an integral component of understanding the somatic aberrations in cancer. So we as clinicians or in pharma or R&D would be remiss if we didn't turn our attention to the recognition that fusions can drive the development of multiple different types of cancer. And then skipping down to the fourth element there, NGS and high throughput sequencing enables systematic discovery of these gene fusions with great fidelity, with both high sensitivity and with great precision. One of the challenges that occasions is that often multiple gene fusions are identified even within a single individual sample, which can present challenges in distinguishing between oncogenic driver lesions and other unimportant passenger aberrations. Things like incidental fusion events that result in chromosomally unstable cancers, otherwise bioinformatic mismapping, challenges between similar regions in the genome that tend to mismap and lead to artifactual fusion events, or otherwise things like read-through fusion events that are adjacent genes that seem to look like fusions that have chromosomal material in the bioinformatic analysis that doesn't belong together, but in fact is a reflection of a bioinformatic artifact. So that makes it challenging to fully recognize which of the fusion events that are identified in any one of these individual patients or in an entire cohort of samples are the actual meaningful driver lesions. So too are chromosomal rearrangements frequently observed in cancer and in benign tissues, again, complicating the analysis just as I described for 5 above. But the benefit of this is that increasingly these fusions can be used to subclassify these cancers to provide diagnostic, prognostic, and increasingly therapeutic information so that clinicians can best afford their patients a high likelihood of cure with some of these precision therapeutics that target these different types of lesions. So this is from a different group, from a more recent review article from Gao et al. In 2018, and I apologize for how busy this is, but I'll describe what is being conveyed here. In panel A, off to the left, each of those rows represents an individual cancer type. Don't ask me what the acronyms are, but you may recognize some of them. Then the colored bars is a reflection of how those patients within that disease cohort stratify in terms of the frequency of those patients who have fusion events as a driver mutation. And then over on the right is a box and whisker plot breaking those different categorizations of patients with different types of molecular lesions. And the arrows that I'm drawing your attention to are those that include fusions. And so you can see how fusions contribute very significantly across a whole patient population to the meaningfulness of their molecular genotype. This is, apologize for how busy this slide is. I won't go through all of the details, but this is meant to indicate how very heterogeneous these different disease populations can be, both in terms of the number of fusion events that occur in these different diseases, as is showcased by the size of each of these bubble plots, where each of the columns now reflects those different disease categories. That's in column A, and then over in column B is a reflection of the great distribution of different types of genes that are involved and at what level based on the size of those individual icons in that sort of heat map. And so again, the details aren't important. The takeaway message is that fusion events have myriad driver genes and come in multiple different flavors in the form of the different partners that they take on to cause disease. And then I think this is my last introductory slide. It's just to drive the point home that increasingly we're seeing multiple different disease types with some significant fraction of patients with those diseases having meaningful driver fusion events. And then over on the right, the good news is that increasingly there are targeted therapies that can focus their attention on alleviating the molecular lesion that's the result of those fusion events. So that's the same paper that I described earlier from Nature Reviews, where on the y-axis, this is on the right now, on the y-axis we have the driver genes, and on the x-axis are the different precision therapeutics that have shown efficacy in targeting these molecular lesions that result from these chimeric gene fusion events. And so this grid work is going to be increasingly broad and deep with a more wide array of targeted therapies that clinicians can bring to their armamentarium in treating their patients who happen to have one of these fusion events. So that's the background. Let's now talk about why Genomenon, why the field is moving in this direction of better characterizing gene fusion events. So this is not just true of structural variants, but it is also true of structural variants, and I want to make sure that that doesn't get lost in the conversation here. But when performing molecular characterizations of patients, these are the questions that are for which answers are being sought by the clinician or the team that's putting together the data. So some of the questions I'll list here are which patient variants are activating and what is the supporting evidence? Another question that can be asked more broadly is what is the landscape of functional variants for a given gene or a gene pathway? And that can be useful in clinical circumstances in building out a pre-configured genetic lesion database to streamline clinical workflows, or otherwise in pharma R&D to better understand at a very broad level the molecular pathogenesis of a disease at the hands of lesions in a gene or a gene family. And two, what are the evidence predicated genes that should be included on a diagnostic panel to ensure that you're not missing some of these patients whose driver is from one of the genes or the gene variants that I had described in points 1 and 2 above. Which variants then should I include on that disease-specific diagnostic assay if it happens to be a more focused assay that is at the variant level and not at the gene level as in point 3? And then increasingly, which variants are targetable using those precision therapies? And so all of these questions can be addressed from empirical evidence, the lion's share of which is patterned in the peer-reviewed, empirically determined medical literature. So this gets, again, to where we are presently or where we were as of last week. These are a couple of the main resources that both clinicians and researchers could avail to understand the fusion gene landscape driving their different cancers. So these may be familiar to you. If you've used any of these 3 or similar databases, then I think your interest will be piqued with what I'm about to show you in the way of the gene fusion database that Genomenon has recently produced. But COSMIC, the catalog of somatic mutations in cancer, has a listing of all of the fusion events that they've characterized that amounts to about 300 at present. OncoKB is also a commonly used tool, mostly in research, and it has a characterization of variants and fusion events in a number of driver genes in oncogenesis. I'll talk to how deep, how thoroughly, and how comprehensive these databases have characterized the fusion landscape in a moment. And the last would be the JAX Laboratory Clinical Knowledge Base, which was launched, I think, a year or so ago. And so these 3 databases collect this evidence by and large from the medical literature and present that evidence through their web interface in the form of a database. And we had heard from our users, and we understood, I personally from my own work, that these databases are a shallow reflection of the full complement of fusion events that have been published in the literature. And I'll speak to that more specifically here in a moment. But the answer to that challenge, the need for better characterized holistic landscapes of gene fusion events in cancer, and the dearth of information, the sort of the paucity and lack of comprehensiveness of the databases that have characterized gene fusion events to date, is what Mastermind is coming to the rescue for in the form of this gene fusion database. So as Candace mentioned, and as I indicated too, this is a new development that arose out of a need that our clinical users and some of our pharma partners have mentioned to us. And so with our great data backbone in Mastermind, predicated on the medical literature that I'll talk to here toward the end of the discussion in terms of how we produce this data, we asked the question of the Illumina TrueSite RNA fusion panel genes, which amounts to about 500 genes total. What are the gene fusion partners for those genes, and what clinical circumstances, including diagnostic or disease import as well as therapeutic significance, do those gene fusion events find themselves in the medical literature? So that was the sole input that we asked questions of Mastermind for relevance to the gene fusion database. On the right, at a very high level, is a summary of the results. So for each of those 500 or so genes, we found between 1 and 95 gene partner fusions. So these are in-frame, characterized fusion events that have been found in patients. So this is not a database dump or a sequence assay of a large cohort of patients where we took all comers. These are thoroughly characterized fusion events that have some merit if you were to see them either in your clinical workflow or if you were to be afforded access to this data to inform some of your research and development activities as a researcher or a pharmaceutical company. In total, across all 507 genes, we found approximately 2,500 gene fusion events. So that is not to say every time we found an EML4 ALK fusion, we counted it as a new fusion event. No, this is unique fusion events found across all of those 507 genes. So that is a great richness of evidence and information for any one of those genes on the list. And as I indicated earlier, the list was chosen to be somewhat of a companion diagnostic for clinical users or researchers who are using the TrueSite fusion panel. But we can repeat the process with any and every gene upon request. But for those 500 genes, we found on average 5 fusion partners amounting to 2,500 total unique characterized gene fusion events. And just to give you a sense for the scale with which this project was undertaken, Mastermind, as you may know, has a great corpus of evidence from the empirical literature, and the identification of these gene fusion events came from a subset of the 6 million plus full text and supplemental data sets that Mastermind has from the literature, amounting to about 30,000 articles. So again, those other databases that are currently in use by clinical labs and research groups, those are predicated by and large from the medical literature. You can imagine the challenge that would be occasioned, let alone in having to sift through the 6 million plus references that may contain fusions, but even once you are aware that these 30,000 references are those that cite these fusion events, you'd still have the massive undertaking of pulling that data out and having it be annotated and characterized. And that's effectively what the Mastermind Gene Fusion Database has done for those 2,500 gene fusion events. So this is a pretty key takeaway from the webinar and why I put it here in the middle of the discussion. This is a more detailed reflection of some of the top genes that were most recurrently associated with fusion event. Many of these won't surprise you. Perhaps though the scale of the promiscuity of these gene fusion partners will. ALK, in this case on the far right, we found to have the highest number of gene partners out of the list of 507 at 95. And then it goes on from there. There's a sort of a shallow slope where many of these genes, I think more than 100 of the genes on the 507 list had more than 10 fusion partners. And I want to put that into perspective, characterizing that in the context of what some of the attendees may already be familiar with, and that is to say the Cosmic database has about 20 gene partners for ALK that have been characterized, again, coming from the literature. So in that situation, there's a 5-fold increase in the extent to which Mastermind's Gene Fusion Database has characterized the literature for these functional fusion events. I don't have the data reflected here, but similar numbers for the other databases. There are 7 fusion events for OncoKB for ALK, which for OncoKB was the largest set of data for fusions. And then for CKB, there were only 4 fusion events that were characterized for ALK. So there's between 4 and 20-fold more fusion events in the Mastermind Gene Fusion database than had previously been available. So as we say, that's why we're so excited to be discussing this new release of data. A little bit of technical detail to talk to how we do this. I suggested earlier that we began somewhat arbitrarily with the 507 genes that comprise the Illumina TrueSight Gene Fusion panel. We first determined all of the gene-gene fusions in the medical literature. So that's a one-liner that kind of trivializes the challenge that we faced, but we had a leg up because Mastermind has been extracting gene information for the better part of 4 or 5 years from the literature. And so we use that as a skeleton upon which to frame the identification of these gene-gene fusion events. We assessed the context of the gene-gene fusion co-mentions, both at the article level, the quality of the article, the specificity with which the fusion event was described in that reference, the diseases that were described in the context of patients who had those fusion events, as well as all the way down to and focused on the sentence level where these gene fusion events were mentioned. All collectively, the fusion events were all prioritized by the strength of that evidence using some proprietary computational techniques. They were annotated for the diseases that I described, thoroughly annotated for therapies, any one of those therapies and a much broader array of therapies than I described in the intro slides. That all comprises the annotations for the Mastermind Gene Fusion database, as well as the breakpoints at the exon or intron level. And so where authors have described what exons from gene A and gene B are juxtaposed, Mastermind in its process, both technical and manual, has extracted all of that information and reviewed it for accuracy. And so I'll end with that we review this data to identify and more thoroughly characterize those fusion events that are functionally or clinically significant. I'm happy to announce that we're soon to be releasing some of this fusion data to our professional users. If you're not familiar with the Mastermind interface, this is one of a screenshot from an internet visit for the Mastermind database. And under your account, in the My Reports section, there will become available in some months' time the gene fusion data to our professional users. So if you encounter a gene fusion event in any of your patients and you'd like to assess whether it's been published in the literature, that would be made available to you through this My Reports feature. I apologize that this is so small. There's clearly a lot of data that we're providing. This is a reflection of the information that comprises the Gene Fusion database in its entirety. And so the slides will be made publicly available after the webinar. I think you'll receive an email from Candace, so you'll be able to look at this in a little bit more detail. But for the purposes of the webinar, let me describe these columns of information in the Mastermind reporter that showcases this manually reviewed Gene Fusion data set. So this is a search for one gene. It's the NTRK1 gene. On the left are all of the gene fusion partners listed in descending order of how extensively they've been characterized in the literature. That the number of articles that cite each of those fusion events is listed in the second column, and all of the references, each of the PubMed IDs that correspond to the mention of these different fusion events is listed in the public PMID list. And then the prioritized references that are most likely to be useful for clinical or research purposes is presented right in the reporter in that bibliographic section. And then the ancillary columns there, the fourth and fifth columns describe the summary of the breakpoint evidence that was extracted from the data, as well as the associated diseases, and then not shown as a reflection of those targeted therapies that are described in the context of each of those fusion events. So this is an example for NTRK1, which is one of the 507 genes. This is another example search on NPM1, which has somewhat fewer results, but nevertheless, you can see there perhaps 1,300 reference mentions to the NPM1 ALK fusion event. Again, a bibliographic reflection of those that are most highly prioritized, and an estimation of the frequency with which different diseases are co-mentioned or co-associated with those fusion events as they're driving oncogenesis. And I draw your attention here not to the very well-known NPM1 ALK, which was found in these other databases, but rather to some of the also ran fusion events that are nevertheless clinically very significant and informative for driving different therapeutic or diagnostic decisions. And so if you can't see that, this is NPM1 TYK2, which has been published in the medical literature 9 times, which you can click on that link through the Mastermind reporter and see those references, again, prioritized based on the cogency of the evidence described in those references. And this is the first paper that characterized that NPM1 TYK2 association from 2014. It's a paper that I'm very familiar with. I wrote it. And the reason I chose this example is not because of my ego, but rather because this fusion event, which, as I've suggested, is very well characterized now in the literature and known to be associated with cutaneous lymphoproliferative disorders, is not present in any of those 3 databases that I described, COSMIC, OncoKB, or CKB. And that's just, as I said, I was looking for an example, and I thought I would find one close to home, and it just so happens that it's not in these other databases. So that's a reflection of how shallow those other database resources are, and how, more positively, how rich and comprehensive the Mastermind database is for gene fusion events. So we're rounding the corner here. I want to spend a little bit of time talking through some representative use cases that some of our clinical users and some of our pharma partners have described to us as being potentially an application of the gene fusion database as we've developed it to date. The first for the clinical users is very straightforward. It's just to say to facilitate clinical reporting for patient information from RNA-seq or genome sequencing data where fusion events have been identified. Which reference has it been seen before? What diseases am I looking at? What are the typical breakpoints? And what can I recommend as a therapy? All of that information can come right out of the Mastermind Gene Fusion database very efficiently. Another use case that we're exploring in more detail with some of our partners is annotation of the Cancer Genome Atlas data set or any other clinical database of data for genomic profiling of patient sets to basically have an orthogonal method of annotating that data to better prioritize which of those molecular lesions are likely to be causative, having been as they would have been in the gene fusion database characterized in the literature previously. On a related note, patient selection for clinical trials. As I mentioned at the onset, we would be remiss if we did not include the identification of fusion events in profiling for our cancer patients. And this can be useful for providing evidence and justification for patient enrollment onto clinical trials, and can positively alter the likelihood of success, because you will have better genetic data to indicate that this fusion is likely to be causative versus an otherwise uncharacterized fusion. And then finally, to uncover novel fusion mechanisms that might inform pharma R&D. So I suggested that the Mastermind Gene Fusion Database comprises those 507 known gene drivers in fusion events. We can take a step outside of that gene set and answer the question for any gene, an entire gene pathway, or otherwise very comprehensively across the whole human genome, and thereby uncover fusion mechanisms that are otherwise lying dormant or undiscovered in the medical literature, certainly not discovered in that aggregated, organized, and prioritized data set that only Mastermind's Gene Fusion Database can provide. So I've talked a lot about Mastermind. Again, I was trying to leave the details for last so that I get the main message out there. But Mastermind, for those of you who are unfamiliar, is a comprehensive database predicated on the genomic medical literature, having a deep understanding of the full breadth of the medical literature. 30 million titles and abstracts are indexed on a daily basis. Of those, we've prioritized the full text content for those titles and abstracts that contain any genetic or disease-related information. So this is the most comprehensive data set driving precision medicine and genomics that is available on the market. And we characterize that information for any one of thousands of different diseases, the entire spectrum of human disease, any gene in the human genome. I'm listing here the protein coding genes because we're talking about gene fusion events. And then we have 4.1 million point mutations and indels characterized in the Mastermind database, to which we are now adding those 2,500 gene fusion events that I've just described, and will continue to do so as we expand the genes that comprise the Mastermind Gene Fusion Database. So I believe this is, if not my final slide, it's the penultimate slide. There have been a series of review articles in the most recent issue of Cell. This is one that was particularly interesting to us from Jay Shendur and colleagues. The PubMed ID is down there for those of you who are interested. But this is from table 4 from that review, and Dr. Shendur is talking about the grand challenges in genome science and genomic medicine. And 2 that just so happen to be right next to each other, which was convenient for boxing, were really resonant with what Genomenon's outlook for the future of genomic medicine is. So the first, Dr. Shendur described the generation of catalogs of clinically meaningful, functionally annotated SNVs in all clinically actionable genes. And Mastermind would like to expand that to include all possible SNVs and indels, as well as copy number variants and what we're describing here, fusion events, so that the full complement of genomic or genetic lesions in all diseases across all genes, not just those that have previously been deemed clinically actionable. That is really what Genomenon's driving mission is, so that we can have a better informed, more efficient routine use of exome and genome sequencing to guide cancer therapies or otherwise diagnosis, prognostic predictions in non-oncology diseases, as well as increasingly some therapeutics for those constitutional diseases. And so with that, I will thank you for your attention again. I will turn it back over to Candace to see if we have any questions, if we have time left over. But I'll leave this slide up for any of you who are interested in accessing Mastermind now for the variant content that it contains in the professional edition, and encourage anybody whose interest was piqued from what I described for gene fusions or other structural variants to reach out to us at hello@genomenon.com or Kiel, Kiel @genomenon.com to reach us generally or me specifically. And I thank you.
Thanks, Mark.
Thanks, Mark. Are you muted? Or is that just my echo? Ah, there we go. Thank you so much for listening, and thank you, Mark. Great job. I'm very excited about this new data. Now let's get some of these questions answered. Let's start with Ron. Ron asks, do you assign temporal weights to rank the scientific relevance of articles based on how recent or how old they are?
That's a great question. The answer is I can make it simple and say yes. The more complicated answer is that is one component of the ranking that we perform for prioritization of the articles. So just to take you a bit behind the scenes, I'm happy to dig in deeper on a one-on-one conversation, but the depth with which the lesion, in this case the fusion, is described, in terms of the number of mentions from a computational perspective, also where in the reference it's described, if it's in the title or abstract, that indicates that the authors and the editors have deemed it of significant focus for that study. And then also how intimately described it is in the context of either a therapy or a disease. And to put that in computational terms, that is term propinquity or nearness. So if a fusion event describes a therapeutic compound and a disease in the same sentence and does so very frequently throughout the reference, that paper is highly relevant to the concerns of a clinician who identified that fusion in their patient, or otherwise to a researcher who'd like to know better about what's going on with this gene fusion event and what are the drugs that can be brought to bear to counteract its molecular effect. But I'll emphasize that it's not pure computation. So some of those proprietary computational techniques that I described, those are only used to organize and annotate the data from our perspective, and then that annotation is used to manually review the information, which is why we're able to have upwards of 20-fold more better annotated information compared to some of these other available databases. So that was a great question.
Okay, Michelle asks, in the report, what does all effect types and all calls filtering do?
All calls is a reflection of what I think it was Ron who asked the question of how do we adjudicate the importance of the information. And so being as the database is set up with a technical process initially, there are some scenarios in which there are false positive results. And then there are other scenarios in which the fusion is a correct fusion that we've identified, but it is not clinically or functionally characterized. And so that category is a way to debulk the information so that you can focus your attention on those fusion events that have received the best quality assessment for being clinically or functionally relevant. Some of those false positives that I'll describe are constructs that have been made to test some empirical aspect of protein-protein interactions, right? That's not relevant for clinical diagnosis, and it's less relevant for research purposes in this context. So those are examples of results that are filtered out in the manual process, although they're technically correctly identified as fusions. Other examples would be GST tags to a gene in a pull-down assay. Well, GST happens to be a gene synonym, and so it would look like to our technical process that is a proper gene fusion, but those are unimportant. Those aren't actually real fusions, and so those get weeded out. Where it becomes useful is if a clinician has this lesion and they want to see any information that characterizes that lesion in the medical literature, we don't want to be paternalistic and filter out that information, but our model in Mastermind has always been to show the evidence that we've collected and let the user adjudicate it very facilely in the Mastermind interface, that's where you would benefit from not having that filter on, is when you want a maximally sensitive view into the data. So that was a great question too.
Great. Christopher asks, does Mastermind have all of the fusions observed in the TCGA dataset or only fusions for driver genes?
Good question.
Good question. So
we have in our database those fusions that have been characterized in the literature. And you'll be aware, if you've spent any time in the TCGA dataset, that it's annotated, but it's largely unexpurgated. So it's a reflection of all of the genetic lesions that were found in these different patient cohorts. And so we do not have all of the lesions, the fusion events in TCGA, but that is beneficial because what we have is a highly focused, empirically characterized data set of fusions that can be useful to filter out some of those read-through or artifactual or otherwise passenger fusions that are found in TCGA. That's not to say that if it's not in the gene fusion database, it's not informative, but it is annotating the TCGA fusion dataset with the Mastermind dataset is a useful way to let those fusions that are most likely to drive oncogenesis to rise to the top, and to do that at scale. But it's certainly the case that any of the fusion events that are in TCGA can be assessed in the Mastermind Gene Fusion database to answer that very question. I'd like to see any evidence in the literature where this fusion has been characterized, or if there's no evidence, then I would follow a different sort of diagnostic chain of events to assess whether the fusion that I found in my patient is clinically meaningful.
Awesome, thank you. Colleen asks, what is the difference between the functional variant landscape you mentioned and the fusion database?
Oh, great. So perhaps I'm being a little bit loose with my terminology here. Variant, in that case, for the functional variant landscape is point mutations and indels. So the data that comprises the Mastermind software at present. The functional database of fusions is strictly focused on fusion events. And so that's the difference, is just the size of the lesion. And the functional variant landscape has a different mechanism of characterizing the clinical meaningfulness. In the case of constitutional disease, we follow the ACMG framework, as we've described in other webinars. And in the case of cancer or somatic lesions, we follow AMP guidelines or any permutation of AMP or similar guidelines that our pharma clients have requested of us. So again, the functional variant landscape is for single nucleotide variants or indels, and it follows those schema, the ACMG and or AMP classification criteria. The functional gene fusion database is a collection of all of these gene fusion events that has then been manually reviewed in ways that we've just described for clinical or functional significance, including which diseases are associated with those fusion events and which therapies can be brought to bear for treatment, and also a characterization of the breakpoints.
All right. You just mentioned breakpoints, so I'm gonna ask Santhosi, and forgive me if I got that wrong, to what level are the breakpoints characterized for the fusion partners?
Great question. At present, we have at the exon intron level. If what you're asking is for chromosomal positions, that information, if it's available in the author's reference, that too can be incorporated into the annotation. But at present, we're staying at a high level, just letting the subscribers to the Gene Fusion database know which exons are juxtaposed. But it's not only a benefit to have identified these gene fusions and to be able to annotate them, but also we provide you with a roadmap for answering some of these more in-depth questions. So suppose that there's a question like you're asking that isn't addressed by the annotations that we've provided. You now have in your arsenal all of the papers where that fusion event is cited. And so it's a very short path from knowing that that fusion event exists to going to those references in Mastermind and identifying what those precise chromosomal coordinates for the breakpoints are. If you're asking, on a related note, if you're asking whether it's in frame or out of frame, that information generally comes out in the author's description of the identification of that variant, and that is included in our manual annotation of the gene fusion database.
All right, thank you so much, Mark. There are several more questions, but I think we're going to wrap it up here. And what we'll do is we'll have Mark answer them in a text format, and we will send a follow-up email with a link to the recording, the slides, and the Q&A within the next week. So thank you so much for joining us today. Hope that you learned a lot. Be sure to visit us at genomenon.com and follow us on all social media at Genomenon, and you can email us at hello@genomenon.com if you have any further questions or want to discuss the gene fusion database further. Thank you again for coming, and have a great day.